Welcome to An Introduction to Health Research, compiled by the RUNX-1 Research Program for the Research Guided by Patients Initiative, funded by a Eugene Washington PCORI Engagement Award for Capacity Building. This training is meant for a patient and family member audience who want to learn the basics of health research. The objectives will be to answer all of these questions about the process, from study creation through sharing the results with various audiences to impact the healthcare system. The ultimate hope of this program is that RUNX-1 FPD patients and family members are equipped with the basics to understand and assess RUNX-1 FPD research reports and summaries, and two, to begin to imagine where they can become more involved in all of these research steps to help the RUNX-1 FPD research field move more efficiently, effectively, and quickly toward better RUNX-1 FPD care options. So let's start with what is health research. Health research is sometimes referred to as medical research or clinical research. Health research refers to the many types of scientific investigations that aim to test ideas, answer questions, improve treatment options, and increase knowledge about human health. Health research is on a circular continuum, starting with population research that helps to identify a problem in a general population, to laboratory research to investigate the problem and possible solutions. This is often called preclinical research. And then on to clinical research to test in humans specific treatments or other types of interventions to address the problem originally discovered. And finally, transferring what is learned in previous steps into new therapies. You might recognize that the NIH RUNX-1 FPD research study, sometimes referred to as a clinical research study or a natural history study, it's considered population research. As you'll learn in a couple of weeks, many RUNX-1 FPD research studies are in the laboratory or preclinical phase. And then finally, our team is working with our research community to design an intervention study based on existing drugs. Currently, we're a little too early to discuss the study, but please stay tuned. Again, the whole health research process is cyclical, and it starts over as new problems and related research questions are identified at each step along the way. The rest of this training program relies upon a really nice PCORI developed training package. It was created by PCORI to introduce health research to an audience like you. PCORI developed it with a focus on interweaving patient-centered outcomes research concepts throughout. For our training, we have set aside these PCORI concepts as best we can because we will introduce those at a later time. This allowed us to condense this 3-hour training program to 1 hour. You will continue to hear reference to PCORI concepts, but really what these modules in this training program will focus on is the basics of health research. The PCORI program includes 5 modules that will walk the learner through the research process. It is really interactive and includes self-assessments along the way. And you are welcome to navigate to the link at the bottom of this page if you'd like to complete the full program or at least walk through these modules in your way and go through some of the self-assessments and activities. Otherwise, as you advance through these slides, you will watch condensed video versions for each of the modules, and you may decide to pause and re-listen to any section you'd like. Now, as you listen, you will hear a range of health research examples. We encourage you to pause and also think about how RUNX-1 FPD questions that you or your family members have can apply, and think about associated studies that may be created at each of these steps along the way and how you might be involved. Now let's start with Module 1. How are research questions developed?
Have you ever asked yourself questions like these? We all ask ourselves questions about decisions we need to make. But what if we don't have the information we need to make informed decisions? When research is patient-centered, it can provide the answers we need to make good decisions about healthcare. Welcome to Developing Research Questions. What will you learn from this module? A research study tries to answer a specific question by collecting and studying information, also called data. In a study about healthcare, data might be things like interviews with patients, records of patient appointments, or medical record excerpts. The process used to guide how data is collected and analyzed is called a research method. Scientific research only uses methods that have been tested and proven to provide trustworthy answers. There are many different kinds of studies. Some can be called trials or clinical trials. Let's look at some questions many patients or caregivers have about depression and see how to develop a question into a research study. It's a major health issue and affects many people. There are many possible questions you might want researchers to answer. These are some common questions people experiencing depression have. Think about which ones seem the most interesting or important to you. Let's look at the ways that researchers try to answer questions like these. The first step is looking at the evidence. What has research already found out? To understand what is already known, researchers use a process called systematic review to identify all the studies that have been done on a topic. During a systematic review, researchers look at what all the studies together tell us about our question. They also look at whether studies were done well and produce trustworthy results. As part of the systematic review, researchers also look at what we don't know, called research gaps. Research gaps let researchers know what still needs to be studied more, or in a different way. For example, could the outcomes of a medicine for depression be different for people who haven't been included in studies, such as people of a certain age, race, ethnicity, or sex? Do the effects of treatment last over time? Were there harms as well as benefits? Once a research team has learned what is already known about a topic, they'll have a better idea about what questions still don't have evidence to help answer them. They can then consider how the gaps relate to questions patients and caregivers most want answered to make informed decisions. Let's review these patient-centered questions about treatment for depression and find out how to answer them using a research study. Asking which treatments work best for people like me is a question that can be answered by patient-centered outcomes research. Often, doctors know that several types of treatment can work for a specific group of people. There may be some studies that demonstrate that two medicines, let's call them medicines A and B, can be used to treat depression, and a different study may show that a kind of talk therapy, we'll call it option C, is also effective for reducing the symptoms of depression as well. But doctors do not know if one works better than others for a specific population. PCORI can help doctors, patients, and others learn which treatments work better for certain populations, such as men or women, or for people who have more than one health problem. To create a PCORI study comparing treatments, researchers make a series of decisions. The PCOTS acronym stands for Population, Intervention, Comparator, Outcomes, Time Frame, and Setting, and outlines those decisions. First, after looking at what research has been done and hearing the priorities of patients and others, researchers need to decide who should be the study's population. To answer for whom, researchers may need to keep in mind that a treatment best for one group might not be best for another. For example, people with kidney problems may need special monitoring if they take medication. Next, what do we want to compare? The research team might want to compare a new healthcare option with an old one. Or they may want to look at what happens when someone gets a test every 6 months instead of once a year. In a research study design, the thing being studied is called the intervention. In this example, the intervention is medicine A. A PCORI study compares the intervention with other possible options, called comparators. Sometimes, all the options are being compared to each other, making the options both interventions and comparators. Next, what should we measure? How do we know if a treatment works? People on the research team might have different ideas about what to measure. A doctor might be concerned about results of lab tests, or whether symptoms have gone away entirely. A person with depression might be concerned about whether they can sleep better. A caregiver might want to see improvement in how many days their partner or child can go to work or school. The results of a study are called outcomes. An outcome may be a benefit, I can sleep better, or a harm, the medicine upsets my stomach. Let's talk a little more about outcomes. Outcomes are important results that might happen because of an action or intervention, such as taking a medicine or increasing people's exercise. An intervention can have positive and negative outcomes. Not all outcomes will be equally important to each person. A medicine might improve a health condition by reducing a symptom, but it might have side effects like headaches or weight gain. Side effects are also outcomes. They just aren't outcomes that people see as positive. Outcomes may not always be related to physical symptoms like pain. For example, an outcome could be how well someone may be able to participate in daily activities. Next, the research team needs to decide how long the study should last. How long should people get the treatment? And how long after treatment should the team follow up to see what difference it made? Every research question is different. In a study about reducing infections during surgery, the results might be clear in a few days. A study about reducing risk of heart disease, on the other hand, might take years, enough time to see if more people got sick or stayed healthier than expected. It can take a couple of months for depression medicine to work, and the team might want to see if patients feel better 6 months or a year later. Suppose a study compares 2 treatments. If it stops at 6 weeks, we will learn something about the outcomes at that point. But results might look different over time. In the illustration, at 6 weeks, the treatment shown by the blue line shows more benefit than the treatment shown by the red line. But at 18 weeks, the red treatment shows more benefit. The setting where the study will take place also is important. If studies have only looked at treatment for patients in large cities, the research team may want to know if results are the same in rural clinics. The research team may want to test 2 approaches to solving a very specific local problem. Or they may want to draw patients from many parts of the country so that the results can be applied to people from many different backgrounds. Now, let's see how all the pieces add up. We just walked through the main parts of a study at a glance using PCOTs. Now we have complete research questions that the team can use to plan the study. Stakeholders contributing to a patient-centered outcomes research study can shape each element of PCOTs.
Now, welcome back to our training. We'll go on to Module 2, Designing the Research Study.
Welcome to Designing a Research Study. I'm Jerome, and I'm a patient advisor on our research team. I'll be your guide for this module. I'm going to tell you what I learned when I helped a research team design a study, and how my input helped the team make some important decisions. Here are some of the questions I had. Just so you have the whole picture from start to finish, a research team usually follows these steps. Identify the research question. Choose a research method. That's the one I'm talking to you about. Identify where the information or data will come from. Collect the necessary data. Analyze the data and determine the results. Share the results with others. First, the team had to create a research question. That was the topic of Module 1. Our research question compared the benefits and harms of different cardiac rehab programs. The next step is to decide what method to use to answer the question. You might want to think about a research method as a blueprint for how the research team will answer the research question. It lays out what kind of data to collect and how to collect it. Methods also help researchers analyze the data or understand what the data means, and then tell the story of the data as it relates to the research question. How a study is designed or what method it uses is really important. Methods add credibility to the study findings because they explain the steps taken to answer the question as accurately as possible. There are three main types of research methods. Quantitative methods use numbers, also called statistics, to measure the relationship between things. Qualitative methods use people's descriptions of their experiences or perspectives to answer questions. Some research teams use both, which is called mixed methods. Quantitative studies answer questions by counting and measuring things. For example, blood pressure readings from a group of patients. How most patients rate their health on a scale of 1 to 10. How many days patients spend in the hospital. For these studies, researchers may use medical records, surveys, lab results, and other sources. Researchers look at the relationships between different things being measured, called variables. For example, a study might want to understand the relationship between taking a medicine and having lower blood pressure. The team looks at how each medicine affects blood pressure. Qualitative methods collect people's descriptions of their experience and study the meaning people give to these experiences. Using the same blood pressure example, a qualitative study would ask people to describe how taking the medicine made them feel and how it affected their daily lives. People might tell the story about how their life changed after they started taking the medicine. Researchers collect those stories from interviews or focus groups. They might also observe people doing something and take notes of what they saw. To answer a research question using a qualitative method, the research team must interpret these responses, looking for similarities and differences between people's experiences or behaviors, and draw conclusions about what the data means. Sometimes researchers choose to combine qualitative and quantitative approaches to create a mixed methods study. The study to compare the benefits and harms of two blood pressure medicines might collect blood pressure readings and other health measurements. They could also hold focus groups with those taking one or the other medicine to talk about if taking a medicine caused them any challenges or difficulties, or affected their life in a negative way. Mixed method studies can help researchers measure outcomes, as well as describe the ways that people think or feel about those outcomes. When I was choosing a rehab program, I had a lot on my mind. I wanted a program that would reduce my chances of having another heart attack. I wanted a program that could help me make changes to my eating habits. I also wanted something convenient, near home or work. A qualitative study could collect people's experiences to see whether the program helped them get what they wanted. A quantitative study could compare the numbers of people from each program who had another heart attack or who didn't lose weight.
In this section, we'll talk about how quantitative and qualitative studies are organized to answer the questions credibly. This is called research study design. Most patient-centered outcomes research studies involve a quantitative component. They compare treatment options using statistical tests of various measures. There are several ways to compare treatment options. One way is to observe the outcomes of people who are already using specific treatment options. This is called an observational study. Another way is to assign people to groups and have each group use a different treatment option. This is called an experimental study. Observational and experimental studies can be set up in different ways. Making a choice about how to design a study isn't easy. A lot of factors have to be considered. Here is a set of terms you're likely to hear when your research team is designing a study. Remember that observational studies use groups of patients who are already getting a treatment option that the research team wants to compare. For example, there may be some patients at a healthcare clinic who are taking one type of medicine to help lower their blood pressure, and another group of patients at the same clinic who are taking a different kind of medicine. The research team can observe the results of both groups to compare them. The research team can even use the normal blood pressure checks that the patients get at these clinics every 6 months as a way to compare which medicine does a better job at controlling people's blood pressure. Let's take a look at some examples of study design using observational methods. We'll get to hear how Jerome felt about the design based on the information he was hoping the study would provide. Here is an example of a retrospective observational trial. Take a look at the study design and then click on Jerome's feedback to read about his thoughts on the study. Now let's take a look at experimental design. In this kind of design, the research team assigns people to a specific treatment. One group of patients gets treatment A. A second group of patients might get a different treatment. Both groups measure results in the same ways, and the outcomes of the two groups are compared. There are many ways that the research team might choose who in the study gets treatment A and who gets treatment B. The research team might assign patients to each group by randomization, which means they were assigned by chance, like flipping a coin. Assigning people at random to receive one treatment or another helps experimental studies avoid the type of bias we discussed in observational studies. Randomizing helps ensure that each group of people in a study is basically alike. But research teams need to plan experimental studies carefully to avoid another problem: making sure the results of the study are generalizable. Results are generalizable when the people who take part in the study are similar to the patient population as a whole. For example, diabetes affects people of all backgrounds, but it is more common in people who are Black and Hispanic. If a study of diabetes treatments enrolled mostly white people, it would be hard to know if the results applied to other people with diabetes. The study results would be more generalizable if participants better represented the whole population of people with diabetes. A research team's decisions to focus their study on a certain clinic, geographic area, or patient group may need to trade off convenience or cost with the chance that the results can't be applied to all patients with the condition. Sometimes the study is designed in a way that keeps patients from knowing what group they are in or which treatment they are getting. This means the study is blinded. Sometimes the study is designed so that even the researchers don't know who is in which group. That is called a double-blinded study. Blinding makes sure that no one influences the outcome because they have an opinion about which treatment is better. And even when researchers or patients try to avoid it, that opinion can change the way information is collected or the way that results are interpreted. Here is an example of a randomized trial design. Take a look at the study design and then click on Jerome's feedback to read his thoughts on the study.
There are a lot of study types to choose from, and I have some concerns. How do I know if a study is going to get accurate results? How long is it going to take to get the answers I want? How do I know if the study applies to people like me?
Research teams need to choose study designs that will produce results that are valid, reliable, and generalizable. Validity, or having valid results, means that the results of a study are based only on the things that were measured and not something else. Reliability, or having reliable results, means that if another research team did the same study with the same methods, they would get similar results. Generalizability, or having results that are generalizable, means that results of the study could apply not just to people in the study, but to other people with the same condition. One type of error is a false positive. A false positive means researchers think they have found a difference between two things, but it doesn't really exist. For example, researchers conclude that there is a difference between medicine A and medicine B, but in reality, what looks like a difference is just the result of chance. Another type of error is a false negative. This means the analysis finds no difference between two treatments, but in reality, a difference does exist. Errors like these can happen for several reasons. The actual test used to measure the outcome, such as a certain blood test, doesn't always give accurate results. The study has too little data, or the results are very different across the people measured. Either can make it difficult to get accurate results from the statistical tests researchers use in their analysis. Research teams can reduce the chances of false positives and negatives by designing a study that has enough people for the analysis they want to do. They can also use measurements, such as lab tests or surveys, that are known to give accurate results consistently. Research teams don't create bias on purpose. There are reasons why a team might feel uncomfortable using random assignment, especially if it means that some people don't get a certain treatment they might want or need, or that some people get a treatment that is not best for them. A research team must carefully consider the tensions between giving people a certain treatment and avoiding biased or confounded results. This takes a good deal of discussion, and the perspectives of both researchers and stakeholders, including patients, are important when making decisions. Designing strong research studies is a complex process. Input from patients, caregivers, and other stakeholders can build stronger studies if their concerns are addressed.
Next, in Module 3, the training is focused on planning informed consent and study protocols and protecting patients' rights.
Welcome to Planning Patient-Centered Consent and Study Protocols. Hi, I'm Louise. I'll be your guide in this module. My daughter Amy was 6 when she was diagnosed with leukemia. When looking at treatment options, I realized how little anyone knew about the best way to treat her illness. I enrolled Amy in a study when she was sick, but I didn't really understand all of what we were signing up for. She recovered, so the treatment worked, but the experience made me interested in working on other studies affecting children and parents. Parents really need to understand what is being studied and how the study involving their children will be done. For a research study to be carried out in a patient-centered way, the plans that guide the study have to be created with the protection and safety of patients and caregivers in mind. Researchers call these plans protocols. Everyone on a research team needs to help make sure research protocols contain all the things the team will do to conduct the study. This includes information about how the rights of people taking part in the study will be protected, like the right to keep their medical condition private, the right to be informed of any risks for harm, and the right to be treated ethically. Protecting the safety and well-being of research participants in the parents' study is really important. With the help of a colleague I'll introduce, I'll be explaining how the overall plan for a study, called the protocol, can do that. We'll also explain the form that research participants all sign, called the informed consent, and what information they should be able to find there. To explain the details of a research protocol, I'm going to hand it off to my colleague, William. William is the principal investigator on the parent study. His explanations of how the team works really helped me become a part of the team.
Thanks, Louise. I'll rely on your comments to provide additional insight. A protocol is a plan for the procedures a research team will follow to conduct a research study. A protocol answers questions like the ones you see here. Who will be in the study? Where, when, and how information or data will be collected, and how participants will be protected. Having all of these decisions written down in the protocol helps keep the research team on track and documents the thinking and planning that went into the study. It also makes it easier to make any necessary changes as the study takes place. The research protocol answers those questions through a detailed plan that begins with the reason for the study and the research question, and then the methods the study will use. The methods are the decisions about the type of study, along with the specific population, intervention, comparators, outcomes, time frame, and setting of the study. All the things that were talked about in Module 1. The protocol also includes detailed information about how the study will be conducted in a section called Procedures. There's also a section on ethical considerations, how the research team will protect the rights of those participating in the study. Here in Module 3, with Louise's help, we're going to focus on procedures and ethical considerations from a patient perspective, so you can see how a stakeholder's experiences can make these elements more patient-centered. Select the button Louise's Comments to see what she has to say. All research protocols must be approved by an Institutional Review Board, or IRB. An IRB is an independent group that makes sure participants are protected from any harm that could come to them from participation. The IRB evaluates possible risks to study participants, such as loss of privacy, protections against these risks, potential benefits of the research for participants and others, the importance of the knowledge to be gained by doing the study. Universities and other research organizations all have IRBs. Before a study can begin, the IRB will review everything in the protocol, from advertisements used to recruit people to join the study, to the way data is being collected, to the research team's plans for how to help people who may experience harm from the study. The IRB reviews the protocol again anytime there are changes, and the research team undergoes training to make sure they know how to keep people safe in research. Why do we take so many precautions? Because history has shown that these steps are necessary to ensure the rights of participants in research studies. In the past, medical research didn't always put the rights of research participants first. Participants, also called human subjects, were not given a choice about being in the study, were not informed of the potential dangers of the study, or were not cared for properly after the study was over. These three famous cases led to many of the rules that researchers now follow. While these cases are extreme, the issues are the same and can still happen today without these rules. The health and safety of participants must always come before the goals of the research. The Belmont Report, released in 1979, is the basis for the rules and ethical principles that guide modern research. It outlines three important ideas: respect for persons, beneficence, and justice. Respect for persons means that people who take part in a study need to understand what the study is about and the possible risks and benefits. They need to be able to decide whether to participate without feeling any pressure. Beneficence means that research studies should be designed with the least risk and most benefit possible for the people who take part. Justice means that studies should not take advantage of one group to help another. Informed consent is a process that every person who participates in a study must complete. The term basically means that a person understands or is informed of the details of the study and gives their permission to have the researchers do whatever it is they say they'll do to collect information from the person. All the information is provided in a document that the participant must sign before any data is collected from them. Researchers call this document a consent form. There are three key aspects of informed consent. First, the study must be explained, including how the research team plans to collect information from the person and how the information is being kept private. Second, the person giving consent must understand the potential risks and benefits of participating that they might experience. Third, people should understand that they get to decide if they want to be in the study and can stop participating in the study at any time. That sounds straightforward, but there are many ways these protections can be overlooked if the study protocol is not carefully designed. Anyone approached to join a research study should be able to review all the details in a consent form. The form should be written at a reading level that makes sense for the study population and in the languages that are most appropriate. In addition, the research team should be available to answer any questions someone has about the study. A consent form should answer these questions. In addition, the form should make it clear that anyone can leave the study at any time without any penalty. Preparing a research protocol and planning to submit it for review by an IRB are crucial steps in the research process.
Module 4 is about samples, appropriate sample size, appropriate sample characteristics to select, how to recruit patients, and how to think about their needs and their preferences as you think about how to retain them through the target timeline for a study.
Welcome to Sampling, Recruiting, and Retaining Study Participants. Throughout this module, you're going to be working on a study about follow-up care for women who have had breast cancer. You'll be asked to participate as a member of the research team as they reach the stage of sampling, recruiting, and retaining study participants. You'll be able to answer these questions. A group of researchers want to compare two screening methods for new cancer in women who have already had breast cancer. A screening method is a test that detects a condition in people who do not have symptoms. These are the researchers on the study team. Scroll over each researcher's title to learn more about their role. As you can see, there's room on the team for more stakeholders who can contribute to making the recruitment for the study as patient-centered as possible. Here are just a few stakeholders that can offer valuable insights to the team on how to successfully recruit people to join the study. Women played a strong role in developing this study from the start. They've shared that women who have had breast cancer said they wanted to know whether mammography or magnetic resonance imaging, two forms of imaging tests, work best for detecting new cancers. This study compares them head-on. Women also said that going for imaging tests often made them anxious about their health until they got results, so they asked to include surveys about anxiety and quality of life to see if women have less anxiety with one type of imaging or another. Before the team can start to recruit patients, we have to agree on who should be included in the study in order to answer the research question. Research studies test treatments or gather information from small groups of people to be able to make conclusions that can apply to the entire population with that condition. Samples are common in everyday life. For example, news agencies talk with a sample of a few hundred or few thousand voters to predict the results of an election, rather than try to speak with every voter. Based on their experience, our stakeholders, including you, can help identify the important characteristics of study participants. A sample needs to do a good job of representing the larger population affected by what is being studied. Our research question asks about women who have had breast cancer. We now have to define who exactly we will recruit to participate in the study. We may want to study a range of women, including subpopulations, defined as groups of individuals who share a common characteristic, such as gender, age, or race. We want our research to apply most to the kinds of women who need to make choices about the imaging they use. Research teams use inclusion and exclusion criteria to be as specific as possible about the people in their sample. These criteria help them choose who should participate in the study. Usually, the sample has the same characteristics as the people the research wants to inform, like people of a certain age. But the sample also needs to leave out people who may have additional characteristics that make them different from the larger population and might confuse the study results. Characteristics of the sample may include being of a certain gender, age, or diagnosis. Making these choices helps to identify which characteristics, also called variables, could influence our results. Here's what our team decided would be our inclusion and exclusion criteria. There were 3 things that we wanted to make sure that women in the study didn't have. These things might confuse the results of the study comparing magnetic resonance imaging, or MRI, to mammogram for feelings of anxiety. One important decision for the team is how many people we need in the sample to get reliable results. This is called sample size. To decide how many people need to be recruited, researchers consider questions like, what are the outcomes in the study? What measures are being used to capture those outcomes? And how likely are women to drop out of the study? Researchers use something called a power calculation based on questions like these. This is a math calculation that tells them how many people they need to study to represent the whole population accurately. Deciding on the sample characteristics to include or exclude has an impact on other aspects of the study, such as the cost of recruitment or the time it might take to find the people who match a large set of criteria. This means the team might have to make a trade-off on one aspect of the sample to keep the study on time and on budget. Now that we know who should be in the study, it is time to find those people, tell them about the study, and engage them in the consent process. Researchers call this recruitment. It's one thing to enroll women to join the study. It's another to encourage them to stay in for the full study, especially one that lasts for years. Keeping people in the study is called retention. Every good study design plans for some people to move away or lose interest and leave the study. That's called attrition. But when too many people drop out, the study results may no longer be meaningful. In this section, we'll be looking at ways we can encourage women to stay on through the end. There are different reasons why people might drop out of a study. Therefore, encouraging study participants to complete the entire study may require several different strategies. Ideally, our team wants to minimize the burden of what we ask of our participants, communicate with them in helpful ways that keep them connected to us, and link them to care if they need it. Making sure that the people in our study get good care is our ethical obligation. We want to make that as easy as possible and tell potential participants how we will help them if they need treatment. Our real-world experience helped to make our study successful by identifying the real problems that could have prevented people from joining and staying in our study.
So finally, Module 5. This is about what the research is complete. What do researchers do to understand the data and apply it to the health system and impact healthcare for patients?
Welcome to Understanding and Sharing Research Findings. In this module, we look at how teams think about the study findings and who can use them. We also look at different ways to analyze data. We'll learn how research teams think critically about what their results mean and what limitations may help shape the results. Finally, we look at where PCORI study results are most often used and how research teams may choose to share the results with different audiences. In this module, we'll be covering these 5 topics. The research team has to use data they have collected to determine valid and reliable answers to the research questions. These are the study's findings. Research findings make a statement about what is probably true for the majority of the population that was studied. So, if a study is comparing one surgical procedure over another, the findings would be true for people who are similar to the people who participated in the study in age, gender, or other characteristics. There are several kinds of findings that might occur in PCORI when we compare 2 or more interventions. Let's take a study of 2 different medicines for lowering people's average blood pressure. Some of the possible different findings are: people taking medicine A had more benefit and less risk. They had greater reduction in their blood pressure than people taking medicine B, and fewer side effects. Or, both medicines reduced blood pressure by about the same amount. There was no difference between them. Both medicines had the same amount of benefits and harms. Or, both medicines gave the same amount of benefit. They both helped lower blood pressure by the same amount, but medicine B had more side effects. When researchers say that their quantitative results are significant, they might mean one of 2 things: statistical significance and clinical significance. Both are important, so let's talk about them both. First, the results of a study are tested to see if they are statistically significant. Statistical significance is a measurement of how much the results are due to the intervention being studied and how much was due to chance. The measure of statistical significance is called a probability value, usually called a p-value. A p-value is a mathematical calculation. It is usually calculated by a statistician. When a finding is statistically significant, it means that people can feel confident that the finding is real and did not occur due to pure chance. Clinical significance means the findings are meaningful and useful to doctors and patients trying to achieve a specific outcome. In other words, the findings show a significant effect. Stakeholders play an important role in determining if a finding has clinical significance by using their own experience and perspective about what makes a finding meaningful. When documenting the findings in a report or journal article, the research team presents the raw results of the data they have collected, and then makes sense of the results and what they mean for clinicians, patients, or other stakeholders. These reports tell a story about how the data collected answers the research question. Here are examples of 2 research reports so that you can see how findings are communicated.
Every study has some kind of limitation. Some limitations are a result of the way a study was designed. Research teams often have to balance the intent of the study with the amount of time or resources available to do the study. For example, a team might not have enough resources to recruit participants from multiple locations, or to recruit from a wide variety of settings that treat populations as diverse as the full population. Or the team might only have funding to track results for 1 year, but the study results would be more informative if the benefits or risks of treatment were studied for a longer period of time. Limitations are a normal part of studies. Research teams must take them into consideration when doing their analysis. They also must report them so others can fully understand the study results. Select the box to see types of limitations that can occur in designing the study. Study limitations created during the conduct of the study may also be due to problems recruiting enough participants or collecting all the data from every participant. PCORI studies increase the amount of evidence available about the outcomes patients care about. Together with results from other research, findings from PCORI studies can help shape healthcare at multiple levels. Policymakers are concerned about the health of all citizens. They help decide what types of research are needed and make policy changes that help people lead healthier lives. Some PCORI studies look at ways to deliver whole programs of care. For example, nutrition classes for people with diabetes, or education for new mothers. Hospitals, health systems, and insurance companies can use results of PCORI studies, along with other research findings, to plan ways to deliver care that can benefit many people. Professional medical organizations use research evidence to develop guidelines on the best ways to prevent and treat health problems. By providing data on outcomes that matter to patients, PCORI helps guidelines committees consider both the benefits and harms of potential treatment recommendations. PCORI results can give patients and doctors, nurses, and other clinicians information to help them choose treatments that best match patients' goals and needs. A recent PCORI study found that there was no difference in average blood sugar levels measured by a test called an A1C for patients with type 2 diabetes who checked their blood sugar levels several times a week versus those who did not. The findings suggest that people with type 2 diabetes who are not taking insulin and whose glucose levels are well controlled may not benefit from checking their blood sugar several times a week. Clinicians see a lot of patients with type 2 diabetes, many of whom don't take insulin. This study suggests changes in how we talk to those patients about checking their blood glucose. Clinicians can lower the number of test strips that patients have to buy and continue to check to make sure their diabetes is under control. The diabetes educators and support team in a health system can use the findings to refine the instruction and guidance given to patients whose type 2 diabetes is under control and who aren't taking insulin. It also gives patients a milestone to reach for as they bring their glucose levels under control. Less testing is a great incentive. The Medical Society's guidelines have never specified different blood sugar testing routines for different patients with type 2 diabetes. Now it is considering whether it is time for more focused recommendations for patients with type 2 diabetes who are under control and not taking insulin. The diabetes educators and support team in a health system can use the findings to refine the instruction and guidance given to patients whose type 2 diabetes is under control and who aren't taking insulin. It also gives patients a milestone to reach for as they bring their glucose levels under control. Less testing is a great incentive. The findings from this study have important implications for the burden patients experience when trying to manage their diabetes. The study opens the door for additional research that could change healthcare policy to support other aspects of self-management, like zoning for healthy food retailers or creating more spaces for movement and exercise. Research findings can't do much good if they don't get into the hands of people who can use them. That's why research teams need to think about dissemination. Dissemination is the intentional, active process of identifying target audiences and tailoring communication strategies to increase awareness and understanding of evidence, and to motivate its use in policy, practice, and individual choices. In other words, dissemination is communicating the evidence to a specific audience in ways that help them understand and use the information to make decisions, improve practices, and create policies. Here are some standard places where research findings are often found. Stakeholders have an important role to play as the research team studies its data and plans how to report its findings. Thinking about what you and others like you might want to know can help inform how those results are shared.
Now that you are familiar with the whole research process, one thing we want to dive into in about 5 minutes is what is a clinical trial? What are the phases? What do they mean? And how does the process work?
When you get sick, do you ever wonder where medicines come from? Behind every pill, injection, or cell-based therapy you might be given lie decades of laboratory research and hundreds or thousands of failed experiments along the way. Once potential treatments are discovered, they are subjected to rigorous testing in animals, and the most promising treatments are then tested in humans to make sure that they are safe and effective. Ultimately, this process takes the innovation and ingenuity of hundreds of scientists together with billions of dollars of research and testing. The treatment's journey from the bench, where it is discovered, to the bedside when it can actually treat patients, has several stages that culminate in a thorough clinical trial. Clinical trials are research studies in humans that explore whether a medical treatment is safe and effective for people. In the US, these studies and the important data they generate are approved, regulated, and evaluated by the Food and Drug Administration, the FDA. The best clinical trials are performed as randomized, double-blind, controlled trials. This means that patients are split into 2 groups. One group receives the experimental treatment, and the other group receives a placebo, an intervention that has no therapeutic effect, or current standard of care. The purpose of this design is to make sure that the experimental treatment itself is responsible for differences, not just the perceived improvement of a placebo. Double-blind means that neither the patient nor the doctor knows what group the participant is in. Before undergoing any treatment, all participants of clinical trials go through a process called informed consent, designed to make sure they understand the potential benefits and risks of experimental treatment, as well as those of the placebo or standard of care. In accordance with the FDA's requirements, human clinical trials have 4 phases that each has a specific purpose. Phase 1 trials which can last up to 2 years, test whether a new treatment is safe and well tolerated, including side effects. Phase 2 trials, which can last longer than 2 years, test the treatment to see if it is effective. Is it doing what we hope it will do? And to further evaluate its safety in more patients. Phase 3 trials are the most critical evaluations and are performed on a larger group of people to understand whether the treatment works to prevent or treat disease better than any alternative treatment. This is called the treatment's efficacy. They typically last 3 to 5 years. After a Phase 3 trial, if the treatment has demonstrated sufficient evidence of positive benefit, it can be approved by the regulatory agency, the FDA in the United States, and can be commercialized. Phase 4 trials are done that monitor safety for any rare or long-term side effects of the treatment. Phase 4 also evaluates the treatment's effectiveness in real-world situations. Regardless of the outcome, participation in clinical trials is always valuable and brings us closer to curing or mitigating disease. In most cases, it takes more than a decade for a candidate therapy to move from Phase 1 testing to real-world distribution. Remember that human trials will celebrate successes, but barriers will surface, along with complications and challenges, so patience and understanding of the scientific discovery process are essential. This is especially true as California scientists work to develop a new generation of therapies, cellular therapies, that will have unique challenges that differ from traditional drug-based medicines. Clinical trials are absolutely necessary to determine that a treatment is safe and that it has a real positive effect on a particular disease, better than that observed by a placebo or the standard of care. Going forward, we must build on the progress that California's remarkable scientists and physician scientists have made. Only with continued support for biomedical research can we bring the most innovative therapies to the patients who need them most. To learn more about clinical trials, check out the ISSCR's A Closer Look at Stem Cells.
Now remember, health research is cyclical. Research questions feed studies to create answers, which eventually feed into new questions and future studies. At any point in time, research will describe what we know to date, but that will quickly lead into more learning that may expand or even contradict aspects of previous studies. Any one research study cannot stand alone for an area of study, so it's important to think of the context and the whole body of literature in that area. The whole health research endeavor has led to longer lives lived and greater well-being, and diseases that were once deadly are now manageable. This process can take decades, but we're doing our best here at the RUNX1 Research Program to make and facilitate the RUNX1 FPD research field to advance as efficiently and quickly as possible. We hope you walk away from this training feeling ready to learn more about findings to date on RUNX1 FPD and then learn how you can become a research partner in each step of this process. Please remember, we cannot do this without you, and we are so glad you're here.